Perfect. All right, it's still my pleasure to welcome you today to the dissertation defense of Morgan Sparks. So Morgan has had the privilege of being surrounded by 3 families while he's been here at Purdue. And the first of these families is near and dear to both of our hearts, theoretically, which is, of course, the salmon family. So this is a phylogenetically correct tree of the salmon family. And when Morgan joined my lab, he had just gotten his master's degree from the University of Alaska, where he had worked on sockeye salmon. So these salmon right here. And he had worked on thermal adaptation and phenology in response to climate change. When he joined my lab, he quickly brought to my attention that there's this really interesting introduction event of pink salmon, which are these guys right up here, which were introduced into the Great Lakes in the 1960s. And so we quickly figured out that he could go up to the Great Lakes and sample them. But more importantly, what he found out is that the Siebes at the University of Washington had tissue samples from the original progenitor population in British Columbia. So we could use the DNA to compare the progenitor and the introduced population. And thankfully, Lisa Siebe agreed to share and be a collaborator with us. And she's also a committee member of Morgan's, and she's on Zoom here with us today as well. So the bulk of Morgan's dissertation focuses on how pink salmon adapted to this totally novel ecosystem of the Great Lakes. Starting in June, though, Morgan's going to again remain immersed within the salmon family, and he's going to be working on these guys right over here. So these are cutthroat trout. And so Morgan has an NSF postdoctoral fellowship to work at Colorado State University and look at patterns of evolutionary rescue and genetic rescue in cutthroat trout. The second family that Morgan's been a part of is, of course, the Christie Lab family. And as you can see here, I think we're doing a pretty much spot on impersonation of this cover art. This is, of course, Abbey Road from the Beatles. But rather than thinking about, so Morgan has been a great addition to our lab and has been a great person to have in our lab. And rather than sort of think about him leaving, I like to think that he's just going to be part of our extended Christie Lab family for many years to come. And then last but not least, Morgan has, of course, extended his own family. So he and Melissa have a son, Harrison, who was born January 16th. And I think Harrison's in the back there. There he is sleeping. So yeah, he's only about 3 months old. And yeah, I'm really proud of Morgan for not only putting the finishing touches up on his dissertation, but also being a committed dad during that time period. So overall, I'm really proud of Morgan, and I'm really happy for him to talk to you about his work today. So I'll turn it over to Morgan. Thanks, Morgan.
I can turn it down like that. Yeah, but not the best. I think that's working all right. Andy, can you see that yet? Or is it just a little bit behind still? Yeah.
Looks good.
Great, thanks, Lisa. Well, thank you everyone for being here today. I'm really sorry, I'm trying to get my pointer up here. There you go. All right. Thank you everyone for being here in person today. I'm really excited to be sharing this research with you, and thank you all that are also there on the live stream. Just so you know, you can enter questions into the live stream chat, and we'll have someone that's looking at it that we can address those questions at the end. And so I'm going to be talking about a portion of the work that I did for my PhD time here. So I'm going to be talking about these fish that Mark alluded to earlier, which were pink salmon that were introduced to the Great Lakes. And in particular, I'm going to be talking about the genomic consequences of that introduction. And so to get us started, I wanted to give us a brief outline of what I'm going to be talking about today. And I'm going to start with a little bit of background. I'm going to move on to the study species and the collection of these fish and talk a little bit about the ecology of these fish in their native and introduced range. And then I'm going to be talking about the population structure and the effects of gene flow in the Great Lakes, thinking then about genetic drift after that, and then rapid genetic adaptation. And actually, I'm going to add another little section onto that where I'm thinking about genetic adaptation in a slightly different way. And then finally, I'm going to synthesize these results and talk about how they relate to some of the concepts that we care about more broadly. And so to get us started, I'm going to jump into this background section, and I'm going to start really broadly. And I'm going to start with this idea of evolution that I care about a lot. And the way that I'm going to define evolution for you today is simply just the change of allele frequencies from one generation to the next. And so as some of you who particularly work with salmon may have noticed, we have a ray troll theme going today. And here's just a ray troll piece of art, thinking about evolution. We've got a bunch of kind of evolutionary icons. We've got dinosaurs. We've got Charles Darwin driving his Volvo. And I'm going to commit kind of an evolutionary biologist sin and lean heavily on this cliched quote from Theodosius Dobzhansky, where he said, "Nothing in biology makes sense except in the light of evolution." But the reason I'm going to commit this kind of evolutionary biologist sin is because it's really true. When we think about all the organisms that we see here that are both dead and alive, or extinct and non-extinct, when we think about the way they looked, when we think about the way they interact with the world, and when we think about the way they interact with one another, ultimately that comes down to these heritable units that I'm talking about today. And so that's why this quote is, you'll see it's featured so heavily in evolutionary biologist talks, but also I think is kind of a fundamental truth in biology. And so when we think about evolutionary processes, I think, especially if you're not an evolutionary biologist, we tend to think about evolution in terms of macroevolutionary processes. So these are the evolutionary processes that take really long amounts of time to complete. So for example, we've got our single cell organisms in the primordial soup that we're representing down here, the rise of tetrapods from ray-finned ray-lobed fishes, and then also the rise of humans out of apes. And so these are the processes that take hundreds of thousands, millions, sometimes billions of years to complete. And I think that's a lot of the way that evolution is framed, particularly in popular media. However, I'm going to use this Gary Larson cartoon and kind of a bit of a Lamarckian joke here, in that we're looking at these little aquatic organisms that are needing to evolve over a single generation to go get their baseball. And so we would consider this a microevolutionary process, or something that takes just a few generations, or sometimes maybe tens of thousands of generations to take place. And typically, when I'm thinking about microevolutionary processes, I'm thinking about them in terms of rapid evolution. And so we can define rapid evolution as evolution on the scale of tens of generations or less. And there are different ways that we can think about rapid evolution. So for instance, we can think about them in the different forces of evolution. So that would be selection, gene flow, mutation, and genetic drift. These are the things that actually change allele frequencies from one generation to the next. And just to give you a sense of what I'm actually talking about, I'm going to use a recent example of selection. And so there was a study that was completed recently that looked at these tusked and tuskless phenotypes of African elephants. And what they were looking at is that these elephants were actually really heavily poached as a result of a civil war in Africa. And so what the researchers were observing was that there is this transition between tusked to tuskless phenotypes in these populations. And they actually looked at the underlying allele frequencies, and they found that as these tuskless elephants became more common, as you might expect, the alleles that underpin this phenotype also became more common. And so this is an example that's 50 years long, but we're talking about just a handful of generations with respect to elephants. And so this is kind of how rapid evolution can actually look. And the way that I would like to propose studying rapid evolution, or I like to think of studying rapid evolution, is with respect to these introduced or invasive species. And I think these would make excellent models for studying rapid evolution. And so we've got our kind of cast of villains that you can see here. Cane toads, starlings, rats, and invasive sea lamprey in the Great Lakes. And there's a couple different reasons why I think that these are such excellent models for studying rapid evolution. So one is these organisms, when they're introduced to a new environment, they've got these novel conditions that they're experiencing. And we expect them to rapidly adapt. So that's a form of selection acting on these in these populations when they're introduced. Similarly, we tend to know something about their introduction story. So we may not know the exact population from where they're founded from, but we do know kind of generally where they came from and when they may have entered the ecosystem. And we know that typically introduced species occurred usually they're occurring as the result of human introduction. So they had to have taken place in the last few thousand years generally. And then finally, or additionally to that, we know that since we're not moving whole populations to new environments, we're taking a subsample of those individuals, there are these effects of genetic drift on these populations when they enter these new environments. So there's these bottlenecks and founder effects. And then finally, there's also these sometimes intense control measures on these organisms, particularly the invasive species in these environments. So for example, lamprey were introduced to the Great Lakes, and they cause a lot of damage to fish that we care about. And so folks in the Great Lakes have implemented these control measures where they design pesticides to specifically target these sea lamprey, and we were pretty efficient at killing them. So we're actually imposing our own form of pretty intense anthropogenic selection on these individuals. And what this allows us to do is to answer questions about our invasive or introduced species, things that we might care about managing through their kind of evolutionary processes, but they also allow us to answer questions about species that may be less tractable. So we can think about rapid evolution in terms of species of conservation concern that are less manipulable than these introduced and invasive species. Or alternatively, we may have species like in the case of elephants or like whales, where the generation time is quite long from one generation to the next, and so it's much harder to study rapid evolution. So the species that I'm interested in looking at rapid evolution in are these fish that you can see here, Oncorhynchus gorbutia. So these are salmon that are in the genus Oncorhynchus, which is the Pacific salmon genus, and these fish were introduced into the Great Lakes. And so the questions that I'm interested in answering for you today, or that I'll be focusing on answering for you today, are first, how do these novel life histories affect gene flow in the Great Lakes? Second, what was the effect of genetic drift on these newly founded populations? And then third, how did these fish adapt to a radically different environment? And so with these questions in mind, we need to think a little bit about the ecology of the fish and the history of this introduction story. And so now I'm going to transition into thinking about these study species and my collection of these study species. So pink salmon are an obligate anadromous fish. And so they're native to the North Pacific, which you can see here. And essentially they're native they're found particularly from around the Puget Sound region in Washington, all the way over to northern Japan. And when I say obligate anadromous, what I'm talking about is this life history for these fish. So these fish their parents spawn in a freshwater environment where the eggs are laid in a red. Those eggs take some amount of time to develop, where those fish emerge and then spend a little bit of time in the interstitial space in the gravel there. And after they emerge from that gravel, typically pink salmon will directly outmigrate into the ocean. So they're moving almost immediately into this marine environment. And it's in that marine environment they actually spend the majority of their lives, approximately 1.5 years, where they eventually return to that generally same freshwater habitat, spawn, and die. And the thing that's really important that I want you to keep in mind throughout this presentation is that this freshwater environment is an environment where there's less available solutes and ions than what's available in their body. So they're actively trying to keep these solutes and ions in their body. But when they go into this marine environment, they're going into an environment that's rich in these solutes and ions. And so they're actually, when they're breathing through their gills, they're bringing in those solutes and ions into their body. So they have to physically move them out of their body. And so this is a pretty extreme physiological change where they're changing their osmoregulatory function. So the second thing that you need to know about pink salmon, which is quite unique, is that these have these obligate 2-year life histories. So we can walk through this example here. And so if we consider a fish spawning in 2017, its individuals or its juveniles would hatch, go out to the ocean, spend 1.5 years or so in the ocean, and then complete its life cycle at the end of those 2 years in 2019, then 2019 to 2021. And the same as if a fish spawns in an even year. So this would be 2018 to 2020, and then 2020 to 2022. And if we extrapolate this out of that over the course of thousands of years, what emerges are these divergent lineages that I'm showing you here. So what this functionally means is that a fish that spawns in a habitat in British Columbia is actually more related to an odd year spawning fish in Asia than a fish that spawns in that exact same habitat in an even year. And so that's very different than the rest of the fish in the genus Oncorhynchus. And so we can also think about this just as a PCA, because that might be a little bit easier to think about. So what you can see here is that if we're grouping these populations, the first axis of variation that's being described is this lineage axis, and then the second one is a geographic axis, so latitude in this case. And so the fish that I'm going to be talking about today are these fish from British Columbia, and then the ones in the Great Lakes that come from this population in British Columbia. And so the introduction story technically starts in 1955 when these fish would have spawned, and then they were introduced into the Great Lakes in 1956, and 21,000 juveniles were introduced. And so these progenitors are from the Lake Ells River, which you can see here, which I'm showing you. This is a tributary of the Skeena River, and the Skeena River is quite a large watershed in British Columbia. And so this Lake Ells River population is quite a large population. Sometimes as many as 1 million individuals spawn in a year in that population. And so back in the 1950s, folks in Canada were like, well, that's a lot of fish. Let's found a new population with them. And so what the plan was is that they transported them over to a hatchery here on the shores of Lake Superior, where they're going to hold them, and then they transport them up to Arctic Alaska, Arctic Canada, where the plan was to create a subsistence fishery. And they did this. They actually introduced something like 700,000 individuals to Arctic Alaska. None of those fish took. However, the hatchery manager in Thunder Bay, Ontario, on the Current River, which you can see here. So here's the Current River. That's where the hatchery was. They opened up the raceways thinking there are no fish left in the hatchery. That connects to the Current River that goes out to Lake Superior, and that was the introduction. And so those fish spread rapidly, first in Lake Superior, but then they entered all the Great Lakes just in a few generations. And within about 12 generations or so, they had observed fish in every single Great Lake. The predominant spawning locations are still in Lake Superior and then the northern Lake Huron and Lake Michigan. And that's kind of where we stand today. The other thing, though, that you need to know about these fish was that the progenitor stock, they were from this odd year population. And if we consider the kind of typical spawning life history, they have this obligate 2-year life cycle. But when these fish entered the Great Lakes, what happened was that these fisheries managers started to find fish that were spawning in even years. And they found that very surprising, as you might expect. And what was going on was that these fish were founded from an odd year population, but they started to mature also at 1 and 3 years old. Now, typically, pink salmon in the Great Lakes still mature predominantly at 2, but a decent proportion of the population now matures at 1 and 3. And if we extrapolate, or if we think about this kind of schematic that I showed you, if a fish is spawning like in 2017, if it matured at 1, it would spawn in 2018. And if it matured at 3, it would spawn in 2020, which is how we end up with these fish spawning in odd and even years in the Great Lakes. And so we can think about this as just kind of a schematic of an evolutionary history. And so these populations are founded from from the British Columbian odd population. And the question is, what's going on basically at the tip of this tree? And so to answer that, like Mark kind of talked about earlier, we said, let's go see if we can catch some fish up in Lake Superior. And so here's the Steel River, which is the study site in the Great Lakes that I used for the study. And then we went up in 2018 and 2019. The first year that we went up there was really kind of an attempt to see, can we actually do this? Can we go and catch fish? Is this project going to be feasible? And so you can see Claire, who was a master's student in the lab at the time, we went up in like the first or second week of October. got pretty cold. It snowed a decent amount. It was rainy. We were camping outside the whole time. We were wet all the time. You can see Claire smiling through the wetness, but we did catch fish, and we said, okay, this project is feasible. And so we went back in 2019, caught quite a few more fish, and you can see these pink salmon that we caught over the course of these 2 years. And then this is a really wild area, so you can see this black bear track there. This place is like it's got black bears coming out of its ears. We saw them almost every day we were up there. So it's a wonderful place to do field work, super fun. And so what this actually looked like, here's a side channel of the Steel River. And what we're doing is we're going in there, we're seining these fish up where they're spawning. There's probably tens to hundreds of fish spawning in the side channel, netting them, taking them over to our mobile sampling station where I measure them, weigh them, and take tissue sample for both genetic data and also to be able to age them. And so what that actually looks like is here are all the fin clips that I took for this project, drying in my garage, very stinky garage at this time period. And what I can do is I can take the bony structures in these fins and actually use them to age these fish. And so my actual sample design for this project then looks like this, where we have populations from the native range. So from here on out, when I talk about the progenitor population, I'm going to be talking about this BC odd population, which you can see here. And then we also included an even year population that spawns in that exact same habitat, which is kind of an outgroup for our study. Similarly, it's experiencing the same kind of environmental conditions that this progenitor group would be experiencing as well. And then from our sampling in the Great Lakes, we sampled the Steel River, which you can see in relation to the current river there. And because I was able to age them, we were able to subdivide them using their year classes. And so we had individuals from that odd year sampling trip, then the 3-year-olds from that odd year sampling trip, which is when we caught the predominant amount of our fish, as well as fish from that even year. And so these were the fish that I actually used for the genomic analyses in the study. And so to do that, we took our sample groups, we prepared individual libraries, and then used whole genome resequencing and sequenced these individuals to approximately 14x. And this is a lot of bioinformatic work, a lot of computer work, very complicated. So it's always important to touch in with your peers and see if you can get their help. And so here I am with Harrison when he was probably 6 weeks old, writing and analyzing the results for this project. So to kind of orient ourselves for what I'm going to be talking about for the rest of this presentation, I wanted to provide a very brief genetics review. And so when we think about how individuals inherit their genetic information, we can see we have our 2 parents here with their sets of chromosomes. They donate each chromosome to their offspring, which you can see here, where they go through the process of recombination. And then, a researcher comes along and samples some of that DNA. And we run it through a modern sequencing platform, which you can see here. This is an Illumina NovaSeq. And what happens is, hopefully you have some kind of genome or assembly resource where you or another researcher has actually assembled the genome for this study species. And so in the case of pink salmon, this is a file that's just 2.7 billion bases long. So it's a very large file. And the key to know here is that when we sequence the genomes of these individuals, what we're not doing is we're not just zipping along the genome saying this base is here, this base is here, this base is here. What the sequencer is doing is actually taking random samples from this genomic information. And then we need to use bioinformatic algorithms to align that back to this resource that we have. So these reads are only 150 base pairs long or so, typically, depending on what sequencing platform you're using. And so we're using these bioinformatic algorithms to actually align these short reads to this very long genome resource. And so we can actually zoom in on our alignment here, and we can look at what's going on in the genome. And so here we have our position right here, the genome resource, which you can see right here, and then the chromosomes from our offspring on the other side of the screen there. And what I'm showing you here is a couple different things. Generally, we're aligning to the same genome as our study species, are a very close relative. So they're sharing the predominant amount of their genetic information. So that's being highlighted by positions 1, 2, 3, 4, and 5, where they share all the same genetic code there. And then what I'm highlighting are these single nucleotide polymorphisms at positions 3 and 7, where we're actually, this is typically the data that we're using to do our analyses. And so the other thing we can do is we can actually genotype these data. And so here at position 3, we have a heterozygous locus, and then at position 7, we have a homozygous locus. And it's these data that we actually use to run the majority of our population genetic analyses. And so, yeah, these are what we're typically using for our population genetic analyses. And so with these data, then what we can do is actually start to answer some of these questions that I posed. And the first question that I wanted to answer for you today is whether or not these odd and even year spawning fish are in the introduced range are differentiated. And one thing I want you to keep in the back of your mind here when I'm walking through this is that we only have samples from a single habitat in the Great Lakes. So I'm not answering this question across all these different populations. I'm answering this question for the single set of fish in one habitat. And so I'll remind you, this is the spawning life history phenotype for the native range. And if we saw that starting to arise in the Great Lakes, what you'd expect to see is we've got a progenitor population and then the odd spawning fish forming in one group and the even spawning fish forming another group. Now, the alternative to that is that we've still got these overlapping years that are sending gene flow from one odd year to an even year and back and forth. And so what that would look like in terms of data would be we've got a progenitor population over here and then odd and even year fish forming a single group as I'm showing you in this figure. And so again, what we're really getting at is what's happening at the end of this kind of schematic tree tip. And so here I'm just showing you the data in that same kind of PCA type approach. And what you'll notice is that here are the colors for all the samples in the Great Lakes. And you can see that they're forming a single group here, which is indicative of there being a single population. Then we've got our progenitor population over here, and then our even year outgroup, which really fits what our expectation is. And so what this suggests is that pink salmon in the Great Lakes, at least in the Still River, form this single distinct group. And why this matters is, so odd and even fish in the Great Lakes are this single population within a habitat. And then these alternative age at maturity phenotypes connect the 2 years, right? It's these 1 and 3s that are connecting the odd and even year spawning fish. And so the thing though I want you to keep in mind is that those lineages that I showed you took tens of thousands of years to evolve. And so this is a real short time dynamic, right? We don't know what these populations might look like well into the future, but we can kind of begin to understand their population structure now. And so this is also important because this has occurred over something like now 33 generations. And so if we think about those 2-year life histories versus the 1 and 2s and 3s, those 1, 2s, and 3s look much more like other Pacific salmon species than pink salmon in the native range, which occurred over a super short amount of time. So then the next question that I'm posed and then we'll answer for you today was, what was the effect of this bottleneck at introduction? And so when we think about bottlenecks, that's a form of genetic drift. And these bottlenecks constitute simultaneous reductions in the demography or the demographic consequences in this population, as well as the genetic diversity. And so what I'm showing you here is a population of fish. What I want you to know is there are 15 individuals in this habitat, and there are 3 genotypes here being represented by the 3 different colors. And so genetic drift as a process is just a random sampling of individuals from a population. So we can do that, and every fish this touches or is inside the circle will be our founders in a new population. And so in this new population, there's been a significant demographic decline. We've gone from 15 individuals to 5 individuals. But similarly, we've lost some of our genetic diversity. This black genotype, for example, is now not present in this new population. Similarly, we've lost a decent amount of the gray individuals. And so I wanted to ask or answer in the Great Lakes was, what were the demographic declines in the Great Lakes, and then the related genetic diversity declines of these fish that were introduced? And to answer that question, first, we'll look at the demography. And I used a program that's called GONE, which estimates a population genetic parameter called effective population size. And that's kind of a complicated population genetic parameter, but we can think of it kind of in a really simplified way as being the number of individuals that contribute to the next generation. And to orient yourself, or what this program does is it estimates these effective population sizes, both for contemporary values, but then we'll project them into the kind of near past. So about 100 generations into the past. And so in this figure, it's flipped a little bit than what you might expect. On the left-hand side of this x-axis are contemporary values. So values for today or when these samples were collected. And then on the right side of this axis are 100 generations into the past. And so what we can see when we look at this is this dashed line is representative of when this blue population was introduced into the Great Lakes. So these orange lines. And what we can see is that these orange lines aren't actually flat, they're just very small. So we can zoom in and see what actually the behavior is there. And the results here are, at the time of introduction, this BC odd population, which is the progenitor, had an effective population size of about 150,000. And that's a very large effective population size, but I'll remind you that this population has as many as 1 million individuals spawning in a single year. So it's a very large population. Now, in the Great Lakes, what you can see is that the effective population size estimate at the time of introduction was very small. It's about 50 at that dashed line. And so GON's a little bit squishy, right? It's not going to perfectly estimate your data. And so you can actually see there's a small decline even before that introduction event. So probably what's going on there is GON's not quite capturing the exact time period of introduction and is just off by a little bit. And so in total, what I'm showing you here is actually a 2040-fold reduction in effective population size, which is a huge demographic decline. The other thing, though, that I want you to keep in mind with respect to these results is these points and the distributions of variance around them are actually the points of these lines, which you can see over here. And so these are our contemporary values. These were the estimates for when these populations were sampled. And what you can see is that, for example, if we look at the progenitor, there's been this significant decline in the number of individuals or the effective population size of this population since that introduction event. And at the same time, these samples in the Great Lakes have actually grown in their effective population size. And that's important to keep in mind because we're going to talk about genetic diversity next. And so since our samples aren't from the period of introduction, they're contemporary, this decline is likely going to coincide with a reduction in genetic diversity, which is to say that our contemporary samples may not actually reflect the full extent of genetic diversity at the time of introduction. And so the next question then is, we have this massive demographic decline. What is the coinciding genetic diversity decline? And so here I'm showing you heterozygosity. If you remember when I talked about heterozygous and homozygous loci, this is just the number of heterozygous sites in your data set for an individual and then summarized as a population. You can see here, or sorry, not the number, but the proportion. And what you can see here is we've got our progenitor population and then our sample groups in the Great Lakes. And just to summarize it for you, what we see is about an 8.2% loss in heterozygosity. However, if we've got some population geneticists here, you're probably squirming a little bit because you know that heterozygosity is not necessarily the best parameter for understanding the effects of bottlenecks. And the reason for that is that when as I showed you in the kind of graphical example, what happens in bottlenecks is you actually end up losing rare alleles. And so we can think about that with respect to SNPs. And so you can see here, again, blue is our progenitor, orange is our Great Lakes sample groups. We see about a 2 to 2.3 million loss of SNPs, which is about 30.8% of those SNPs. Now we can also look at this a slightly different way. And here I'm just showing you the number of SNPs averaged across 2.5 megabase pair windows, which is 2.5 million bases. And part of this, what this is doing is it's actually controlling for some genomic features. For example, centromeres have really low genetic diversity, so we're controlling for that feature here. And this actually shows about a 37.7% loss in the number of SNPs in these windows. So about a 38% loss in genetic diversity. So why does this matter? Well, I've showed you that there's a significant demographic decline, about a 2040-fold reduction in effective population size. And then I'm also showing you this evidence for a large reduction in genetic diversity. So an 8% reduction in heterozygosity, as well as a 38% reduction in the number of SNPs. And why this matters is, so pink salmon are still in the Great Lakes, So they've gone through this both demographic and genetic diversity bottleneck, but we know they're still persisting. And this is important because when we lose genetic diversity, or when an organism or a population loses genetic diversity, this is the currency for future adaptation. And so that's what I'm going to be talking to you about next. How do these fish adapt in the Great Lakes despite this reduction in genetic diversity? And so we can think about this with a few set of predictions, just given what we know about the fish in the Great Lakes. We know that there's these novel life histories. Presumably there's some kind of genetic underpinning for these life history differences. We know that they're experiencing new pathogens in the Great Lakes. So there's this new kind of disease environment. There are different food webs in the Great Lakes than there are in their marine environment. And then, like I've alluded to earlier, they're treating these Great Lakes as surrogate oceans, which is a wholly freshwater environment. And so there's this big osmoregulatory difference in the introduced range than in the native range. And so to answer this question, I used what is called a genome scan approach. And this is a bit of a complicated figure, but you can see these are the chromosomes for these sample groups that we're comparing here. This is, when you're doing this, you're comparing sample groups or populations. So here we have our progenitor and then the Great Lakes odd sample group from this study. And then we're calculating a statistic called FST. And so FST is a statistic of relative genetic differentiation. And actually these points here aren't SNPs, they're windows across the genome. So there are 100,000 bases at a time. So each point here is actually 100,000 bases. And then we have our red points, which are our outliers. And the way that we're calculating outliers here is since we're standardizing this method, so it's actually not FST, it's ZFST, it's just a Z transformation. It's 5 standard deviations from the mean here. So pretty large difference between the kind of general trend in the genome relative to these outlier windows. And in total, there are actually 47 outlier windows that I'm showing you here. And I'm going to only focus on a few of those, in part because you just don't have enough time to talk about 47 windows with multiple genes in them. And one of the ways that I winnowed down these genes to focus on is I used a program called SNPEffect, which actually annotates the function of the SNPs in these particular genes. And I focused on genes that had missense variants. So they're actually changing the amino acids in these regions. And so what I'm showing you here is actually the SNPs in these windows. And then the black ones are outlier SNPs, and then the gray ones are just the not outlier SNPs. And so for example, this gonadotropin releasing hormone receptor had one missense variant. And as we can understand a little bit of the functional information about this gene from work in other organisms. So it's associated with the regulation of puberty and particularly mammals. So changes in this gene are associated with either they're associated either with early maturation or late maturation in mice and humans. We can also move over here to chromosome 24. And one of the things I want you to focus on, this is actually a little bit more zoomed out than the other figure. And that's in part because what we can see here are these big gaps in where our SNPs are. So they're not evenly spread out like they are in this region. And so part of the downside of this approach is that you can kind of get false positives because you have these big gaps in your windows. And just a few highly differentiated SNPs can make it look like you have an outlier. However, there were actually 3 missense variants in this particular gene. And so this is a CD209 antigen-like protein, which is active in the immune defense against many pathogens. And so this gene actually has been linked with combating the pathogen that is responsible for schistosomiasis. And we know that that is actually fairly common in the Great Lakes. The last region that I'm going to be talking about then is over here on chromosome 2. And this is a period circadian protein homolog. And the first thing that I really want to draw your attention to is the SNPs in this particular gene. If we look at the SNPs here, there's 38 SNPs. Almost every single one of them was an outlier based on our differentiation metric. And what that actually looks like is we can look at the allele frequency change in the progenitor population with respect to the introduced populations. And we can see that in the progenitor population, they were fixed at one particular allele. And then when they went into the Great Lakes, they actually became fixed at almost at the alternate allele for that particular SNP. Same is true for the alleles that they had low frequencies, they became high frequency alleles in this introduced population. So this is very strong evidence for selection at this locus. And what does this locus do? I think it's a pretty interesting gene. So it's part of the master regulator of an organism's daily clock. And so what that actually means is that the protein product here is actually becomes phosphorylated, and it helps an organism determine its day length, right? So a normal phosphorylation, an organism would associate a day with being 24 hours long, less phosphorylation, a shorter period, and then more phosphorylation, a longer period. And so again, with all these genes, I'm not saying that this exact trait is under selection. We can't know that necessarily about pink salmon, but we can make informed hypotheses about what to study next and how these are affecting the phenotypes in the Great Lakes. And so one of the ways that I did that is what you're looking at here is the hours of daylight a fish would experience in the Great Lakes relative to the progenitor population over the course of 2 years for a fish in the Great Lakes. So what you can see here is in this period, they'd be experiencing less light, and then like over here, they'd be experiencing more light. And that's because these populations in the Skeena River or the Lake Ells River are quite a bit farther north than the populations in the Great Lakes. And so why this actually matters is, for example, when these fish would be emerging and out migrating from the interstitial spaces in the gravel in a particular stream in the Great Lakes, we can see that this is a period with actually quite a bit less light. And so we know that's important for this life history period in salmon, in particular when their juveniles are emerging from the gravel. They typically do it in the dark because they're avoiding visual predators. And so again, this is kind of a hypothesis. This isn't the actual mechanism for why these fish are experiencing selection, but definitely a ripe area for future study. So why does this matter? I'm presenting evidence for selection in numerous genomic regions, 4,700,000 base pair windows. I'm also providing strong evidence for selection in genes associated with puberty and pathogens as well. So these match some of our a priori predictions, as well as this daily circadian clock, which was not part of our a priori prediction. But if we link this with what I actually talked about in the last section, what I'm showing you is that there's this rapid genetic adaptation despite severe reductions in genetic diversity, right? That genetic diversity is the currency for selection for adaptation for these fish in the Great Lakes. And so we've got this big reduction in genetic diversity in these fish, yet they're still adapting in this novel environment. So the last section that I'm going to be talking about then is thinking still about rapid genetic adaptation, but we're going to be thinking about it with a slightly different genomic mechanism. And so I'm talking about a supergene in the Great Lakes. And when you hear the word supergene, we're actually talking about our chromosomal inversions. And so we're going to do a little bit of introductory chromosome evolution, chromosome biology to kind of ground ourselves in this. And so here we have an individual chromosome. It's going from the 5' to 3' direction, which you can see here, indicated by this arrow. And we've got a little haplotype that I've indicated with this yellow box. And inside that haplotype are a set of genes, which you can see with those red lines. And so if we move from one generation to the next, what actually is happening with the chromosomal inversion is exactly what it sounds like. That haplotype here becomes inverted and is inserted in the genome in the opposite direction. And so what's key about this is that if we follow this genomic region through the course of multiple generations, what's happening is on this chromosome, there's recombination happening, but this haplotype or this inverted chunk is being protected as a result of this inversion. And so what it's doing is it's maintaining the physical linkage of these co-adapted genes, or presumably co-adapted genes. And now that we have the genomic resources to look at this, we're seeing that these chromosomal inversions are associated with really important life history differences across the tree of life. So one example, which is one of my favorites, is in rainbow trout. And so these are the same species. They might even be part of the same population. And so an inversion underpins the life history difference here between a fish that becomes anadromous and goes out to the ocean, like I was talking to you about earlier, versus a fish that stays in that same habitat and is a resident. And so this is an inversion on the 5th chromosome in the rainbow trout genome. But there's lots of examples in monkey flowers and walking sticks and birds where these discrete life history differences are associated with these inverted genotypes. And so in the process of my work, I was looking kind of just at some really broad genome-wide patterns. One of those here is heterozygosity. These are the chromosomes. And you can see in this kind of highlighted polygon, there's this kind of anomalous region, a big dip in heterozygosity. And so that is kind of initial evidence that there's something interesting going on there. And one of the things we can do, what I was doing, or what I pointed out with that kind of cartoon of the chromosomal inversion is that we've got these sets of alleles that are co-occurring with each other in a particular region of the genome. So we can use another population genetic parameter, which is linkage disequilibrium, to really investigate what is happening in these particular regions. So linkage disequilibrium is just the physical co-occurrence of alleles in a population. And so that's actually what I'm showing you here. This figure is a little bit complicated, so I'll walk you through it. It's broken in half. And so this triangle on the bottom is representative of the progenitor population. And then this triangle on the top is representative of whatever sample group I'm showing you above it. And then this is subsampling a bunch of alleles in these populations. And so this value here, the purple, is the R-squared value, or the correlation coefficient for these particular alleles. And if we walk through all the different sample groups in the Great Lakes, what you can see is there's these strong blocks of physical co-occurrence that are not present at all in the progenitor population. We can also compare the two populations from the native range with one another, and we see that there's this lack of the same linkage blocks that I'm showing you right here. And I should say, so here, what I'm showing you is the leading arm of chromosome 10. I'm only showing you a subset of this chromosome arm where that kind of anomalous drop in heterozygosity was. So this is, again, more evidence that there's a chromosomal inversion occurring there. And what I did then next was I used a program called ImClust, which uses a ordination technique, multidimensional scaling, to try to genotype this locus. And what it found was an inversion that was about 20 million bases long, or 20 megabases long. And so that program can actually genotype the individuals that you give it. And what you can see here is that blue is a non-inverted genotype. So there's no inversion in that genotype. And then green and salmon are either heterozygous or homozygous for that inverted genotype. And so, and you can't count up all these different bars. We're actually showing the kind of probability of these genotypes for each individual here. So I've done that for you. And what you can see is that these samples in the native range don't have the inversion. And then only 2 samples in the introduced range also don't have the inversion. So the rest of them are either heterozygous or homozygous for that inverted genotype. So what that functionally means is that we should consider these native range populations as non-inverted, and then these Great Lakes samples as either being heterozygous inverted, which I'm showing you here, or homozygous inverted. However, if you did some quick mental math when I showed you my sample groups from earlier, what you'll know is I only have 134 samples in this study. And so that's a bit difficult to think about the full evolutionary history of this region. And so what I did is I actually went back into the literature and looked at other studies that had genomic data, whole genomic data for pink salmon in the native range. And so some of these data come from Christensen et al., which is actually the genome assembly paper where they did some whole genome resequencing for different populations. So we had populations from British Columbia, northern Japan, and then our lab also has an unpublished data set of fish from Alaska, which is this Prince William Sound data set. And so I genotyped that same region on chromosome 10 using ImClust, and I genotyped 1,038 individuals in this analysis. Not a single one of those individuals had the inversion genotype. And so what this tells us is that across this really broad range in the native range, basically the whole extent of it, and quite a few samples, there doesn't appear to be this inverted genotype at all. And so that tells us either that we know that this inverted genotype in the Great Lakes seems to be fixed or nearly fixed, either heterozygous or homozygous. And so either what happened was this inverted genotype was captured in the bottleneck and then likely rose to fixation as a result of a soft sweep in the Great Lakes, or alternatively, it evolved in the Great Lakes and then rose to fixation as a result of a hard sweep in the Great Lakes. And so we can start to understand the answer to that question then by thinking about what is functionally going on in this inverted region. And so the way that I did this is I used an approach called gene ontology, where gene ontology just allows us to annotate the function of the genes in our region or in our genome. So we can annotate either like the molecular function or the biological function, as it pertains to the rest of what we know about genome biology. And so what we did is we annotated all the genes, 40,000 plus genes in the pink salmon genome, and I used an enrichment analysis approach where I compared these functions within the inverted region relative to the rest of the genome, looking for functions that are overrepresented in that inverted region. And I'll remind you, one of the things that I talked about was that there are these big osmoregulatory differences between fish in the native range and fish in the Great Lakes, because these fish in the Great Lakes are treating this invaded environment like a surrogate ocean, but it's fresh water, right? So again, native range fish are where they're spending the majority of their adult lives in this marine environment, where they're actively trying to move solutes and ions out of their body. Whereas these fish in the Great Lakes are in this freshwater environment and they're actively trying to keep solutes and ions in their body. And so when we looked at the gene ontology, the results of this enrichment analysis for this inverted region, what we found was that there's an overrepresentation of osmoregulatory genes. There were 2 sodium potassium ATPase pumps, which is what you'd find in the gills of fish, as well as 4 voltage-dependent L-type calcium channels. So these are also channels that would be transporting ions. And finally, there was also a taurine transporter, and taurine is just kind of a generally important osmoregulatory molecule. So why this matters is, first, I'm showing you evidence that pink salmon potentially rapidly adapted as the result of into this wholly freshwater environment as the result of this putative inverted segment. But also, now that we're starting to be able to look at the sequence data from these inverted regions, we know that a lot of these phenotypic differences that we find are fairly ancient. And so these inverted regions have evolved a long time ago, and selections acted on those inverted regions, and we have these very distinct phenotypes. However, while we don't know the exact age of this region, what it suggests is that because this inverted region has switched from being more or less not present in the native range to being fixed in the introduced range, it seems that these fish are rapidly adapting to this freshwater environment on the back of this inverted region. So with that, I'm going to kind of conclude and summarize some of these results for you and think about how they relate to what we think about with population genetics, ecology, and evolution. So again, I was interested in answering 3 questions. The first was, how do novel life histories affect gene flow in the Great Lakes? And what I found is that age classes have not diverged as they have in the native range, meaning that these 1- and 3-year-old fish are still acting as vectors of gene flow between odd and even years. So they form this single distinct group in whatever habitat you'd find them in, or excuse me, spawning habitat. Additionally, I was interested in what was the effect of genetic drift on this newly founded population. And I presented data that suggested there was a large demographic and genetic diversity decline for these fish in the Great Lakes. And then finally, we know since there's this decline in genetic diversity, we were also interested in how did these fish adapt to a radically different environment. And so what I presented to you is that there was rapid genetic adaptation via multiple loci and genomic mechanisms in this novel environment. So when we think about this as a whole, what I'm trying to emphasize here is that pink salmon rapidly adapted and persists via multiple mechanisms, despite these severe demographic and genetic bottlenecks, right? These fish have lost a lot of genetic diversity when they went into the Great Lakes, but they have still adapted. And I want us to think about this in 2 kind of separate ways that are related to one another. One is invasive species or introduced species, particularly invasive species. We care a lot about controlling those species because they cost billions of dollars every year. And so what this suggests is that we can't just focus on loss of genetic diversity as a mechanism for controlling these species, at least with respect to this kind of scenario, right? We can't say, oh, we only put a few individuals in there, they can't persist. Obviously they can. However, the flip side of that is kind of a good news story for populations, for example, of conservation concern. So we know that either populations that are experiencing declines now, or we know that additionally in the next 100 years, these populations are going to lose more and more habitat. They're going to become isolated from one another. As a result, they're going to lose genetic diversity, experience their own bottlenecks. If we can limit that to a short duration, like if we can connect these populations, they can experience these short-term bottlenecks where they do lose genetic diversity, they do become isolated, and they can adapt to changing environments. But we need to reconnect those populations because this persistence is only going to occur over a short period of time if we don't allow them to eventually regain some of that genetic diversity. And so with that, there's a ton of people to thank. I'm not even going to be able to cover everyone on this slide who contributed to this. First and foremost, I wanted to thank my committee for their help on this project. So Dr. Gribskov, Dr. McNichol, and Dr. Sieb. As Mark said, the Sieb shared their samples with this project, and we would not have been able to do it without the progenitor population. Thank you, Mark. It's been a joy to be in your lab. It's been great to learn from you and collaborate with you, and I've had a ton of fun with my time here. Also, thank you to the Christie Lab. There's a few folks here today, but also past and present. It's been super fun to collaborate with you and work with you and share our successes and our failures together. And I couldn't have asked for a better lab to spend the last few years. Particularly, I wanted to thank Claire and Julia for their help in the field. I could not have gone around and chomped around in bear country for 2 weeks without their help. And they were real troopers out there in the field. And then also, thanks to Avril for her help with all the code on this project. She really paved the way for some of these analyses. Other folks that were super helpful were the Upper Great Lakes Management Unit with the Ontario Ministry of Natural Resources and Forestry. So basically we showed up in Thunder Bay and we emailed with them a little bit. They got our permits like last minute and they were super, super helpful. And then, you know, a couple of the folks there were like, oh yeah, we can take you out to the river. Let's go catch some fish. So they gave us basically the place on the Steel River to go to find fish, which was super helpful and really showed us around Northern Ontario. Tony Kennedy helped actually age some of the samples for this project. Philip San Miguel did some of the library preparation, which was immensely helpful. And then also Chris Christensen led the Genome Assembly project and suffered through a bunch of my questions about that assembly resource. And then funding for this research, I was supported by Peter and Mary Sue Vosser and their fellowship. And I can't thank them enough for supporting me, but also supporting the graduate students of EEB. And then I was also supported through fellowships from both Purdue and then Purdue Biology. Finally, I can't thank my family enough for their influence on me. So my parents are here and you can see them in the picture here. I wouldn't have cared about fish. I wouldn't have cared about the environment. I wouldn't have cared about asking questions about the environment and those fish if it weren't for them. Also, at the back of all this work is my wife, Melissa. We've been together since before I started my master's degree. We've moved all across the country and she has been with me that entire time and I would not have been able to do this work without her. And then finally, as Mark said, we added a new family member in the last part of this process and he's been an incredibly fun wrinkle to the end of this process. So with that, I'm happy to take any questions. You can submit questions if you're on the live stream with on the chat there. We'll take a couple of them and try to answer them. Also, if you want to follow up, you can send me an email, which I have there, or hit me up on Twitter. Thank you. Other questions on these? Okay. Just keep an eye out and let me know if there are questions. Do other folks have questions?
Okay. So you said at the very beginning that it's expanded into all the Great Lakes. And still in all the Great Lakes? And does that include like Lake Ontario?
They did, yeah. So when I say expanded, they've been caught on the high seas in every Great Lake. Whether those fish are spawning like in Ontario, it's hard to know. Part of the problem is that pink salmon, since they're not causing a lot of ecological destruction, we don't care about, or fisheries managers tend to not care about them as like an invasive species in the Great Lakes, but they're also not super abundant and not stocked like Chinook salmon or steelhead or something like that. So they're not a huge sport fishery also. So they're kind of they don't pay that much attention to them in the Great Lakes. So there's not this like big sampling effort to find them. I expect there's probably a few remnant populations in those Great Lakes, but there are definitely lots of fish in like Northern Lake Michigan, Northern Lake Huron, and in Lake Superior. So for example, like the Steel River, they did some mark recapture studies in like the 80s, and they estimated those populations to be 100,000 fish or more potentially.
Doing why the effort for pink salmon on Canada did not work?
I don't know. That's a great question. I don't think they really followed up. It's hard to say. It's kind of part of that paradigm of introduced species to some extent, is that they were putting them in a very similar type of habitat they would have been in the native range, right? And sometimes those species don't do so well in that like exactly similar habitat. And it's when they go into this more different habitat they actually take off, right? And so we see that with lots of invasive species where at home they're doing really poorly, and in their invaded habitat they're doing super well, right? And so maybe that's part of the story there. Also, there was a lot of moving fish around and just saying, let's try it, and maybe not the most ecologically like informed approaches. And so that could be part of it as well. Maybe we hit, like, for example, like steelhead were introduced in the Great Lakes in the 1800s, right, Mark? And those never took off because they're introduced at the wrong life history period. That's correct, right? Like that's the, yeah, okay. And so that could be part of it, right? We're just introducing fish in the wrong parts of their lives, and so they're experiencing too much mortality as a result. Andrew.
Yeah, so this might be maybe my misunderstanding, but with this inverted haplotype, because none of the progenitor populations have that, is there an idea that you have of, is this like positive selection in the new population, or maybe relaxed selection from being introduced from a deleterious variant?
So I didn't show you some of the data. I can might have it here. Sorry, my cursor is like extraordinarily laggy, but so we actually do have some metrics of selection that I didn't show you. But for example, this is extended haplotype heterozygosity. And this red line that you can, or pink line and brown line, which you can see there, are indices of positive selection, which suggests that it probably is positive selection in the Great Lakes. And then also we actually looked at DXY, and you can see that this region has lower absolute differentiation than the rest of the chromosome at that region, suggesting that it's more newly evolved. So there's this combination of being newly evolved potentially, as well as positive selection in that region.
Do you think if they were brought back and retransplanted, that it would then be deleterious to the marine system?
That's a good question. So one of the things that we don't know is that if there are actually inversions in these populations that we discovered with karyotyping in like the 1980s, we don't, because translating karyotypes to assembled chromosomes is very difficult. We don't know if those chromosomes that are karyotyped are the exact same chromosomes that we're looking at today, but they're approximately in the region of length that you'd associate with like chromosome 10 in the assembly, because it's from longest to shortest essentially. But I think that this probably, this inversion is in low frequencies in that region. It's just, what's maintaining that there is hard to know exactly. Other questions? Jeff.
I think that one of the problems with inversion is that they tend to select recessive deleterious. Do you see any of that? We're in the process of trying to get better sequencing information in this region. It's hard to say. If this is a newer inversion though, then the opportunity to correct those alleles is more limited, right? As opposed to something that evolved 1 million, 2 million, 10 million years ago. But it's hard to know exactly right now. We're hoping to have some long read data here soon where we can really dig into the sequence data in that region. Andy, you had a question?
Some online questions, also about the inversion region. Have people documented like similar effects for other anonymous species that have been introduced and now live entirely in freshwater?
I don't know of any inversions that associate with anagamy. I guess sort of that inversion on OMY 5 would be associated with that, whether that's a osmoregulatory thing or if it's a like just a migration phenotype. It's hard to know. One of the things that's really difficult with any inversion is actually linking it to the explicit function. Most of the way that we discover inversions is actually because they're associated with these very distinct phenotypes. So the things that we can visually just say, okay, like in the example of rough, satellite males have one genotype, whereas like the dominant males have another genotype. So we can just say those two birds are very different from one another. Let's look at the genomic differences. So knowing the exact function and what genes are under selection is really hard, in part because these inversions are inherited as a single haplotype, right? Selection on one locus there can sweep that entire haplotype to fixation, despite there being, in this case, hundreds of genes in that region.
More on the inversion, can you like date the inversion with your data set or?
So there's a paper in Current Biology that uses mutation rate and DXY, which I'm showing you here, to kind of get an estimate for that. We need a good estimate of mutation rate, but potentially we could do it with these fish. I've tried it with a handful of mutation rates, just using a range, and they go from like 20,000 years ago to hundreds of thousands of years ago, but that's a very rough look. And so we could use some other methods. There's some other papers that do that, I think more with long read data. So maybe that's something we can do moving forward.
Right, so we're actually 11 minutes over. Yeah, please join me again in congratulating.
Thank you, everyone on the live stream. Thank you for joining. Hi, Lisa.
Later.
I think what we'll do is we got to walk over to another building. And so
Okay.
maybe I'll just shoot you an email when we are set up over there.
Okay, you thinking half an hour, more or less, or what do you think?
Yeah, probably a little bit less than that, but
Okay, well, I'm not going anywhere, so,
short walk, but we'll see. So
okay,
yeah, at least 10 minutes, Mark says. So
okay, well, yeah, you don't need to rush it. I mean,
okay.
take a breath.
Thanks, Lisa. We'll see you in a little bit.
Yeah,